Hello. Welcome to the Science Scribbler Q&A and Classify. My name's Trisha, and you might recognize me from previous streams. And today we're not going to classify together, but we are going to talk about classifications from the project Synapse Safari. So I'm joined by the Synapse Safari team, Juan, Guillaume, and Chloe, and they're going to introduce themselves, and we're going to have a little bit of a chat about the project and how it formed. So whoever wants to go first, they can go first.
Hi, I'm Guillermo. I am a scientist in King's College London in the Centre for Developmental Neurobiology, and I've been studying this for a very long time.
Chloe, do you want to go?
Okay, yeah. Hi, I'm Chloe. I'm a PhD student at the Rosalind Franklin Institute, and Synapse Safari is a project I developed as part of my PhD project, which is to look at the developing mouse hippocampus.
And hi, I'm Juan Burrone. I'm a professor at King's College London at the Centre for Developmental Neurobiology and also at the MRC Centre for Neurodevelopmental Disorders. And I've been interested in studying the brain in many forms, and now more excitingly also with the RFI and with Chloe and with Guillaume doing all of this analysis that we've done here.
Fantastic. So let's start off with kind of a little bit of an origin story. I'd like to know from all of you how you got into science. How did you end up here, and why you're particularly interested in this area of research?
Should I go?
Yeah.
Okay.
Yeah.
So it's been a long path. I actually didn't train as a neuroscientist. I studied biochemistry as an undergraduate, and then it was only in my PhD that I switched to neuroscience, and that's when I really discovered the beauty not only of neuroscience but of the synapse. And it was there that I actually worked on a very specific type of synapse in the retina. These are called, it's a synapse formed by bipolar cells, which have a massive synapse. And the beauty of that is that it's about 5 to 10 times larger than your typical synapse that we have and the ones that we've been studying with Chloe and with Guillaume. And that gives you an unprecedented resolution and access to something, to a synaptic compartment that you can then study in much more detail. And so I did my PhD on that. Then I moved to do a postdoc in the US, and there I was interested not only in understanding the process of how the synapse works, like I did during my PhD, but also the whole process of plasticity of the synapse, that is, changes that happen at the level of synapses. And that's a critical aspect of the synapse, because of the brain, in fact, because it's those changes that allow us to store information, allow us to learn, for example, right? Even though our neurons don't regenerate later on in life, and they are quite static in many other aspects, the synapses are the places that actually change quite dramatically, particularly during periods of learning and memory and so on. While I was doing my postdoc there, I also studied these synapses, but in the much smaller synaptic compartments that we find in the brains of mammals. And then after I finished my postdoc, I then came back to the UK, and that's when I started my own lab here at King's College London. And here I continued to work on these synapses, but I now shifted to working during perhaps the period where the most change happens in the brain, and that is during development. The whole idea that neurons begin to extend massive cable-like structures, dendrites and axons, that then contact each other and form connections with each other. So during this period where neurons are wiring with each other, it's highly dynamic. And so that immediately attracted my attention, because I was interested in looking at things that are constantly changing, and this, which we call plastic in the field. Now, of course, this is because it's such an active region where so many things are changing, things could also go wrong. And so there's a whole number of neurodevelopmental disorders that we are particularly interested in studying as well, that happen during this period of circuit wiring, during this period of high change in the environment. And we've been studying that in many ways and using many techniques. Now, in terms of this project that we're studying here, as I said, these synapses and the arrangement of these synapses and the distribution of these synapses are something that we're particularly interested in looking at throughout the lifetime of an animal or an individual. But these synapses, as I said, are very, very tiny. And so we can't typically see them under the typical light microscope. And so we needed to start to employ other techniques which electron microscopy affords, which is essentially to have a higher resolution imaging approach. And using electron microscopy, we've now been able to start to create these very large scale reconstructions of areas of the brain across development to try to understand how it is that this wiring process is happening as the animal grows and develops. And so I guess that's where this project comes in, feeds in directly to what one part of our lab is doing, which is understanding the wiring of the brain under different conditions.
Fantastic. A very interesting journey then. So I have one question just on that, which is that you say, you know, these synapses are now so small that you can't see them under a light microscope. The ones that you started on, those ones that were quite big, could you see those under a light microscope?
You could, and they were particularly beautiful. So if a typical synapse in our brains is about, say, 1 micrometer, that's a millionth of a meter wide. The synapse that we were studying was about 10 times that size, it was about 10 micrometers roundabout. And the resolution of light microscopy is just under 1 micrometer. So by having such a large terminal, we could now look at the process of the transfer of information at the synapse from one neuron to another in a lot more detail, just simply because we were dealing with something that was essentially massive. I mean, it was such a giant synapse, but they're unusual. That's the issue with them. They're very nice and they're lovely to study, but they're unusual. They're not your typical synapse. When I moved then for my postdoc and the synapse we're studying now, I moved to the more typical conventional type of synapses that our brain have, with all the problems that brought, of course, because now we were dealing with something that we couldn't really see as well.
Wonderful. Fantastic. And what about you, Guillaume? How did you get into this?
I actually also started my life as a biochemistry, and I've actually had a career that spanned many different species. So I actually, my first project was working on plants, and I worked in Drosophila, so flies, and I worked in like a lot of different systems. But one thing that I was always very interested throughout my career is on the synapse. And one thing that always fascinated me on the synapse was where the function of the synapse is related with the structure of the synapse. So from the first studies where people started looking at synapses was actually in the neuromuscular junction. So where a neuron actually communicates directly with the muscle. So it's not your typical synapse, but was the first synapse where people were able to study properly. And in those studies, there were some events that people could measure electrically, but didn't really understand very well what they represented. And it was really, so we're talking on the 50s, 60s, so this is not my career, but when I was learning, was one of the things that fascinated me, is that you can actually, when you start being able to take electron microscopy pictures of these synapses, you're able to start seeing some very small structures that I guess we're going to talk about more and more over this process, called synaptic vesicles. And when people found this using electron microscopy, they understood what the electrical signals were. And it's possible to link the electrical signals with the morphological signals on the synapses, because of the fact that you can calculate the shape, and we can actually have a detailed image of what's happening, where these synaptic vesicles are full of neurotransmitters, and when they fuse with the membrane, they release neurotransmitter, and then receptors on the other side of the synapse will give the electrical signal that was recorded. So this link between morphology and function is something that always attracted me on the synapses. And then when I did a postdoc related with learning in the hippocampus, where I came here to the National Institute for Medical Research, we were starting to see that the signals from synapses also become bigger, and we were able to study in that time, and we found that more synaptic vesicles were on these synapses as well. So there's this link between structure and function that is really very interesting in the synapses, but it's always been difficult because it's very difficult to obtain electron microscopy pictures, but it's even more difficult to analyze them. So
Absolutely.
the first time the synaptic vesicles were identified was like 62 or 63. But from then on, there's been very few quantitative studies because they're very difficult to find. And that's what, like this project, when Chloe joined and started, it became possible to start identifying them at scale, which is something that's never been possible before. And we're very excited for how this going to work out in the future and the kind of findings that we will be able to find.
Wonderful. I guess, Chloe, let's go to you. You've come from like a slightly different path, and I know this because we kind of work together. But tell me how you ended up kind of in the world of synapses from your original starting point.
Yeah, so I actually started out as a nuclear engineer, which is pretty atypical for our current field, I guess. I originally worked on nuclear reactors, but then I realized not many countries are still building nuclear reactors anymore. So I wanted to do something that has more real life impact. And biomedicine is like a great field for this. So I did a master's in radiation biology that kind of connects the nuclear parts with the bioscience as a nice transition. And before I started my PhD at the RFI, I did an internship at a brain machine interface company, and which got me quite interested in the brain. And so when I started at the RFI, every one of us got handed like a huge project booklet with the description of all kinds of projects that we were allowed to choose from. And I saw this project was the synapses and the developing hippocampus. I thought, wow, anyway, I want to do this one. And that's how I got here.
Fantastic. Well, I think it's a good time to talk then. Let's talk in detail about synapses and how they're formed, what the structures look like. And I think we've touched on, why is it so hard to get these high resolution electron microscopy images. I don't know if Guillaume, do you want to find that video that we were talking about?
Yes. So
should I put the video?
Yeah. Yeah. So I guess we can put the video on first. Before we start playing, we can just have a look at it.
Yeah. Can you see that?
Yeah. There we go. Fantastic.
So basically, so I mentioned electron microscopy before, and electron microscopy is a very powerful technique that allows us to see in very, very large detail structures throughout the body, even materials and everything. But in the context of biology, one thing that has always been very difficult is to see the structure in 3 dimensions. So 3 dimensions are very, very important for biology. And so what it's been only possible in the last maybe 20 years is to be able to do this regularly, where you can actually have the resolution of electron microscopy that it's really, you can go to the structure of single molecules, but in 3 dimensions. But the problem is that if you just look at one of these sections, it's very difficult to understand what's going on. But if you look at the structure in 3 dimensions, you can actually start to gather details that is only possible using the 3rd dimension. And we've obtained these pictures probably over 10 years now, and we've done a lot of analysis on them. And one of the first things we were able to do with these images is to actually identify individual neurons from the mess of all the neurons around, because they're all packed together very tightly. And so if we can play the video now, we can see that what we would done here over months and months of work was to manually trace along this over 100 sections these 3 stretches of neurons. And there's in green, you have the axon, which is the bit that transmits information from one neuron. And then with the blue and the magenta, you have 2 receiving neurons, which are independent neurons that receive the information from this green axon. And what you can see is that on these neurons, there's these structures called the spines that receive the synapses. And that at the synaptic site, you can see these balls, these yellow balls, which are the synaptic vesicles that I mentioned before, that are filled with neurotransmitter and are basically tasked with transmitting the information from one neuron to another. So we've done a lot of work on this. As I said, manually doing this, tracing these neurons. We traced maybe 10 to 20 neurons on each of these blocks. But this takes for a very long time, and we were stuck a little bit before this project on what's inside these neurons, because as you can appreciate from those big blobs of material, the information there is mind-bogglingly big, and it's very difficult to extract. So you can see from the different sizes between the synaptic vesicles and the neurons that we were kind of stuck, and we couldn't know how to analyze these images manually anymore, because we had kind of reached a limit. And that's where Chloe's help was instrumental, and the Zooniverse as well.
Yeah. And how long do you think it would have taken to, if you had asked one person, just one person, to create these 3 traces, how long do you think that would have taken for this one data set?
Yeah, I think so. So basically, for this level of resolution, I think it'd be fair to say that if one person was working from, say, 9 to 5, it'd probably take them at least 2 months, I would say, to be sure about all the connections and to have a, I mean, you could do it very, very quickly, but then you'd have too many mistakes. So I would say that probably just the analysis part, because actually one thing we didn't take, I actually was involved more on the acquisition side of the images and the processing of the tissue, and that on itself would take over, I don't know, maybe 2 or 3 weeks of work just to obtain the images. But certainly just for this bit of the analysis, I would say that a couple of months is probably reasonable.
I think it's also fair to say that there are multiple levels at which you can analyze and segment these images. So here, for example, as we've zoomed out a little bit, I hope you can appreciate that we've got these dendrites in blue and magenta and this axon, and we can analyze them morphologically at this very zoomed out level. But then as we begin to zoom in, and you'll see that now as we go closer and closer to the synapse, there's a lot more detail that we can get at that's really fundamental to how the synapse actually works. So for example, as Guillaume was saying, on this presynaptic side, which is the green blob here, there are lots of yellow vesicles. Now, if you imagine that each synapse contains 100, say, vesicles per presynaptic terminal, and that a given neuron will receive in the order of 1,000 or more synapses, and that there are billions of neurons in the brain, then trying to reconstruct each vesicle within even just a few synapses is a lot, essentially. And so what we did up until Chloe helped us was to provide the more zoomed out general morphology and distribution of synapses. And that already told us a lot, because we were interested in trying to understand if there was any logic, any order in the distribution of these synapses along these vast dendrites. So you have to imagine that a neuron has a cell body, and then it has these very long dendrites, and it's receiving thousands of synapses from other neurons that are all communicating with it. And when you actually look at it zoomed out, it looks like a mess. It looks like everything is chaotic. And we've always been interested in trying to uncover if there's any systematic arrangement or order in the system that we can detect. It turns out, and I wouldn't be saying it if it hadn't been this way, that there is some order. And in fact, we found some really interesting distributions and properties in the way that synapses distribute along these dendrites, along the length of these dendrites. But we were lacking detail. We were lacking it was a very broad description. What was going on with the vesicles that are key to how the synapse actually works? Can we actually map them? Can we add them to our very broad distribution maps of the synaptic? And that could then convey more information on the properties of synapses. And it can. The other thing that we're also missing, and which Chloe did an amazing job with, was to also reconstruct the other major organelle that neurons use, particularly synapses, and that is the mitochondria. The mitochondrion is the organelle that one of its main basic functions is to generate energy in the form of a molecule known as ATP. And these synapses are very thirsty for energy. They use up a lot of energy. In fact, neurons are one of the cells in the body that use up the most amount of energy in our bodies. And so mitochondria are fundamental to their function, fundamental really to their function. And so that's the other thing that she's now being able to add. And we can go into that in more detail, I think, in a second. She's not only added the vesicles, she's also put in these mitochondria, revealing all kinds of weird and wonderful distributions and arrangements.
Yeah.
Fantastic.
Yeah.
Yeah. Also,
so
I'll carry on. Go on.
I just wanted to say that another interesting aspect of this is that we've taken a lot of images like 10 years ago, and we're still going through them. And a lot of the analysis we did was just with one of the time points. And there's other time points that we haven't actually scratched the surface of this. And having automated methods allow us to go back and analyze and mine the data that we already obtained, but also it has the added incentive of making us acquire more data, because if we know that we can analyze them, we can devise experiments and acquire more data, which is something that we had almost given up because we thought that we couldn't analyze even the data that we've acquired already, and there's less incentive to acquire new data.
Yeah.
I think that's a problem that exists kind of across a lot of biology, that there's, huge data sets that we're producing nowadays with really, really powerful microscopes. And actually looking at all of what they contain in great detail would take a huge amount of time. And that's why, for example, as Science Scribbler, we turn to citizen science to help us analyze this data, because, as you said, it would take months, if not years, for one person to manually go through and kind of tease out all of the information that's there. But it's also why we really so interested in using AI as well to help us with that, and to be able to segment in these data sets and hopefully create models that can then be used on new data sets, so that in the future that process is really, really fast and you can get the information that we really, really want from it.
Yeah, no, I think that's a great aspect of the work that Chloe has done also, because it's like a new model as well.
Yeah.
Because people normally think that AI as a thing that works for itself, but having this back and forth between AI and people is something that Chloe's work has inspired me at least. Yeah.
Fantastic. So I think it's time to look at the what we've been waiting for and waiting to show, which is the actual results from Synapse Safari. So Chloe, if you're ready, I can share your screen if you're happy to go.
Yeah, sure.
Alrighty.
So I guess I can show first what the mitochondria and synaptic vesicles look like in our data set. Should I do this?
Yeah, perfect.
Okay. Yeah. So here the yellow and blue outlined ones are examples. This is more of a straightforward, like a clear example of a mitochondria. It has a dark outline and then some structures inside, and it's mostly circular or oval. But then we also have some atypically shaped mitochondria that can be very long, and it could have like a restricted region, which we'll see in the result later. So for human eyes, it's pretty easy for us to identify mitochondria once we've been taught how to look for one with a 10-minute tutorial. But still, even the best AI still struggles with finding these structures. And for synaptic vesicles, they just look like these small black circles, but the problem is that there can be so many of them. Like Guillaume said before, it could just take hours to even segment all of them in one synaptic terminal. And this is what the best AI that we have here, the result of how it segmented the mitochondria. This is MitoNet, and you can see it for the longer mitochondria, it gets fragmented. And then even for the smaller ones, sometimes the boundary is inaccurate. And sometimes when the mitochondria is smaller than usual, it would miss it on one slice. So that's why we developed this citizen science project for volunteers to proofread the AI's results.
Yeah, and I should add that for someone that's not a specialist, and actually for some scientists as well that are not trained in biology, people would accept these mistakes as being not important. But if you can imagine that we use this to build detailed models of how these things happen, some of these errors might just creep up and become bigger and bigger. So it's really important, because also, it's impossible to proofread this accurately if we go to big data biology. So it's really careful analysis of the results of AI. It's really important and fundamental for getting correct models of complex structures.
Yeah, I think maybe that was the reason that's why a lot of the special mitochondria morphology was missed by previous researchers, even though a lot of people have looked at mitochondria. But without a complete, accurate segmentation, we wouldn't know that mitochondria actually behave this way during the early development. Yes, and for synaptic vesicles, we also try to first using some computational methods to segment them. There's no AI model available for it, but we used something called template matching, which is you provide a template, which is like a smaller image, and you try to match it over a large image. This partially works because synaptic vesicles are they have uniform size and shape, but also as you can see, the result is quite noisy. It picked up lots of wrong ones and then emerged lots of vesicles together. So this also needs a lot of proofreading before we can use this data. So this is the premise of Synapse Safari, where we do proofreading on Zooniverse instead of just asking volunteers to segment from scratch. And using volunteer results, we got the segmentation that we used to answer biological questions, and we could also use the data to fine-tune the deep learning model that we used, and we could possibly develop new models in the future. And to proofread mitochondria, we had 3 workflows on Zooniverse. They're called correct mitochondria, mito inspector, and mito spotter. We did it this way because we wanted to maximize efficiency. Instead of asking volunteer to correct everything, we first did a feature-based grouping. So some of them we computationally decided that they have low reliability. So we asked volunteers to draw over the original segmentation outline. And for others, we think they have high reliability. So we only ask volunteers to answer a yes/no question, which they can answer in a few seconds on Zooniverse. So here's how the project looked on Zooniverse. I think this is the mito inspector workflow, where volunteers just look between these 2 images with the segmentation and without, and then they can answer is it correct or not. And once they are familiar with this interface, they can really just answer this question in like 5 seconds. And this is for the more low reliability results, where volunteers would be editing the AI segmentation outline with a drawing pen on the Zooniverse interface. And for vesicles, we have just a single workflow where volunteers mark the wrong and the missed vesicle segmentation that detected with template matching. And because in some synaptic terminals, there can be like 100 vesicles, which could be too hard for one volunteer to mark all of them. So we also asked them a question, are all of the vesicles marked? And if not, they could provide an estimate as like it's just to exit this subject, but still providing some information.
I have to say, if I can interrupt, that because I've done segmentation of this sort for so long, I thought it was really refreshing the way the Zooniverse presented this challenge, because it's difficult to make it interesting. We've had a lot of students in the lab, and we always found it difficult. And the way the Zooniverse platform works in terms of making this easy to do and fun to do, it's really exciting, and I found it really fascinating.
Yeah, we had people in my family competing for who could pick the most vesicles or what.
I honestly, like, anytime I was free, like on public transport, I'd go to one of the Mito spotter workflows because you can just swipe left and right and I'd just
Yeah.
get through them. Yeah.
So
Yeah,
great.
no, it's really satisfying to do.
Yeah. But it was also because we really tried to put to break down the tasks so each little task is not intimidating. It's fun to do, it's quick to do, and we try to make it as engaging and not challenging as possible on Zooniverse. So this is a plot to show volunteer contribution. So we launched in 2024, June 2024, and within I think the first 3 days, we got a ton of segmentation. That in 2 days, volunteers completed the Mito Spotter and Mito Inspector workflow, which was really unexpected, and we had to release new data sets sooner than planned. And in the end, we collected over 300,000 classifications from volunteers, and collectively this translates to over 1,700 research hours. And I have a animation here showing the progress of volunteer classification. So this is like the 3D space of our data sets, and each dot presents a subject that is
Beautiful.
like where it is in 3D. And for each, I think that this is from the Synaptic Vesicle workflow. So in that workflow, we show the same picture to 10 volunteers before we stop showing it to other people, to other volunteers, because we think that's when they'll have reached a consensus. So here the color shows the percentage of completion, like how many out of the 10 volunteers have looked at this picture. So when it's all green, it means the entire data set is completed. So you can see how fast it grew from nothing to all of these pink ones, and then how they turned green very fast in 20 days. Because the vesicle workflow is a huge one. Yeah, this is already much faster than we expected.
Yeah, I have to say, as somebody who had never done this before, had never worked with these kinds of sites before, I was very skeptical that it was actually going to work. And I guess as soon as we put the first data set out there and things moved so fast, and we in fact run out of data because everybody had already analyzed all of our data set within a few days, as Chloe just mentioned, that's when I began to realize that we were onto something, and that this really could be a game changer. And it turned out to be exactly that.
I think that's an experience shared by a lot of people who encounter the Zooniverse for the first time. But the community is just so incredible, and people are so interested in contributing to as many projects as possible. We have really, really committed volunteers in our community, even to the point, some people are doing 100 hours in a single project, which is crazy to me. But like they're very interested in contributing to science in general, and it's like really, really powerful. Yeah,
wonderful.
It is yeah. And it's and again, I think you did a great job of presenting it on the platform, because as I mentioned before, we had experience with people, PhD students or master students or undergraduate students, come to the lab and do some of these projects, and it's probably about 50-50 the people that actually do anything, or they just get bored and don't actually do it, even though it's like their job to do it. So it's quite difficult to motivate people to do some of these things.
This is definitely a game changer for us.
Yeah, on Zooniverse, we also we also sent out like newsletter and results updates periodically to keep the community engaged, to keep people interested in this project. And so like here in these two cases, when we uploaded new data sets or released some results, we got a spike in participation. Or like when we went to outreach events, offline, we also get a spike in the participation.
Yeah, I don't know, like when Chloe talks about how we launched the project and then within 3 days the data was finished. It was really, really great, but also we had launched just before a really big campus science festival, and we had to upload data really, really quickly because we'd run out of data to show everyone who was coming to visit the institute. Which was fantastic, but also like it was quite funny at the same time.
Yeah. It's a great problem.
That's a great problem to have, exactly.
Yeah.
Yeah, that was right before my annual leave. I didn't think that I would be doing work, but I had to really accelerate the work to release it before the event. So should I show some results from like the examples of volunteer answers on Zooniverse right now?
Yeah, why don't you show the slides that show kind of the height maps and how you got to that final consensus?
Yeah, okay. Yeah, so yeah, this one should be the more interesting one. So here's an example of where the MitoNet didn't do a good job segmenting the mitochondria. And we show this image to 15 volunteers who corrected the segmentation. And most of them did a really good job in correcting the outline. We can see 2 people didn't actually do anything. Maybe they just skipped the image. And we can see some of the corrections have some mistakes, like this one on the second to the right on the bottom row. But then overall, we combine these results by adding them together and then applying the thresholds to the pixels of. And for instance, if this pixel is included in more than half of the volunteer outlines, then we accept it. If it's less than half, then we don't accept it. So, and this is the final result of these 15 responses combined. And it's a really good segmentation of this mitochondria that the AI cannot segment. Should I show the one for vesicle as well?
Yeah.
Yeah. So yeah, this is an example where the vesicles were really clustered and template matching really missed a lot of the vesicles. And volunteers, they put markers on the missed ones, which is the orange marker here, and then they put markers on the wrong ones, which is the blue markers here. And because we have multiple volunteers working on this, so we did a clustering. So all of these closely packed volunteer annotations would be combined into one. And this cleaned up the volunteer annotations, and this is the final synaptic vesicle detection, which is also a pretty good segmentation. I feel like for one person, you would get like really fatigued when you try to find out what is the center of this vesicle and what is the center of the neighboring one, because they're just so closely packed. But when you have the wisdom of the crowd, everybody put the markers down and then you cluster it, it gets a really clean result.
Yeah.
So would you like me to talk about the fine-tuned model a bit, or like the final or showing some visualization?
Let's look at this visualization, because I think this is the thing that's so impressive, that you know, this is something that was generated by kind of crowd consensus and is kind of, it shows like a work volume that is, as we've discussed, kind of pretty impossible to do as a single research group.
Yeah.
Yeah, and we're just showing 3 here, but I think we got maybe like tens of thousands of these photons segmented with both mitochondria and synaptic vesicles, which is really a lot.
Yeah.
And this really shows a little bit as well. I mean, it's beautiful, but it shows the heterogeneity that exists within these presynaptic terminals. So of these 3 presynaptic terminals that Chloe reconstructed here, there are some that have mitochondria. There's one that doesn't have mitochondria. There's one that has 3 mitochondria. So, and we really don't know what the differences are of these, mitochondria containing presynaptic terminals versus those that don't, or the ones that have more mitochondria versus those that have fewer. And so this is all new data, essentially, to try and understand how it is that the synapse is organized when it does or it doesn't have this massive powerhouse that's meant to give it all its energy. And that's, I think, what's really fascinating.
So let's talk about this data, because has anyone kind of really looked at it in this detail before, or is this kind of new for exploration for you?
So basically, I think people have been able to look at this, but on very small numbers, because it's been done by hand mostly. So people have done 1 or 2 or 3 examples, maybe. So very small numbers of examples, hand-picked. One thing that people have found in the literature by doing small numbers of these things is that the volume of the synapse, the size of the synapse, is generally a very good indication of the activity levels that it does, whether it communicates more or less efficiency. There's also things related to the plasticity, and when people have looked at the number of vesicles, which is very, very few cases, people have found that the number of vesicles normally correlates also with the size in general. But there's very few details, and it's all very broad things. So there's some hints that this is important information, but I don't think that's been done in a large scale before.
No, and I think one of the things that we can do here is that not only do we have many synapses, but also we can place them in a bigger context now. So we can place these synapses along these vast dendritic structures and begin to see if there's something systematic in the way they're distributed in that way, right? The properties, whether they do or they don't contain mitochondria along these dendrites, and begin to analyze at the level of each individual ones of these presynaptic boutons, the distribution of these vesicles within a single synapse, when there is a mitochondria and when there isn't a mitochondria, and so on. So there's a huge amount of information that we really haven't tapped into. And in fact, there's a lot of analysis that we still need to do with this very rich data set. This is a fabulous problem to have, but this is an incredibly rich data set that we now have to try and analyze, to try and get our heads together. What does it actually mean? Because I think, and we may talk about this eventually, I don't know if I'll have enough time, but eventually we need to then try and look at this functionally in some way as well. So we have this structural description of these vesicles and mitochondria within a presynaptic terminal, but what does it mean for the functioning, for the process of synaptic transmission, for the process of releasing neurotransmitter onto another neuron? And that, I think, is where we should probably take this next, right? Which bring in the structure and the function of the synapse together.
The other thing that is unprecedented that we have, and we haven't talked enough about it, I feel, is the developmental side. So a lot of these studies have been done in mature systems, in quite old ones, but we now have these early developmental time points, which are truly unprecedented because people have not typically done a lot of electron microscopy in development, and certainly it's not been done in 3D or with this level of description, in this systematic description. So the fact that we have this developmental side of it is another
really
thing, because we really only know how synapses develop when things are cultured in the lab. So we have very little understanding of how synapses develop in a living organism. So,
and I think this is what Chloe is showing here, no?
Chloe?
Yes.
Yeah.
This I'm showing this 7-day-old mice mouse hippocampus. We segmented the mitochondrion in it, and it doesn't look like anything that we've seen before. It's very branched and twisted, but it's just a single mitochondrion.
And I think it's worth mentioning that like MitoNet, which is the model that we used originally, that we then had our citizen scientists proofread, has been trained on like a huge amount of data to try and find specifically mitochondria. But it didn't perform well on these cases that look so strange.
Right.
And probably it didn't perform so well because it hasn't been seen before, so it didn't learn that they existed, right? So looking at this kind of new data is really, really exciting.
Yeah, there's no doubt. I mean, when Chloe first showed me those very odd arrangements, I had no idea. And these are even more baffling, I think, which deal a little bit more with this location along a dendrite kind of thing. Chloe, you might want to explain these more.
Mm-hmm. Yeah, so our data set, we have, they're from two regions, one distal, one proximal, meaning that one is further away from the cell body, one is closer to it. And surprisingly, the mitochondria in these, in the dendrites of these two regions look very different. So on the proximal side, it's more like what we would expect. We expect the dendritic mitochondria to be elongated and basically follow the dendrites, so they appear tubular. But on the distal side, in both ages, we find these weird beads on a string structure. So you have like a bulging part and then a restricted part. I have a video here that would show it better. So this is tracing this whole mitochondria, and you can see it become very thin, but it's actually still the same one. And then it becomes larger, and then it becomes thin again. So this has been observed in some other tissues like heart muscle. It has been observed in the brain as well in Alzheimer patients, but I think we were the first to discover that there's actually a distance related a characteristic for these beads on the string morphology.
And to tell you the truth, we don't really know what it means.
Yeah, it's really exciting.
This is something, it's completely unexpected in a way. What is it about the mitochondria that, what is it about the structures, these odd structures of mitochondria, distal dendrites? What does it tell us about what's happening in the distal dendrites? What does it tell us about how mitochondria are operating in these distal dendrites? We really don't know.
So that's the next step then, isn't it? We've got this huge library now of annotated data generated by the community on the Zooniverse, and now it's up to you guys to go and work out what it all means.
Right. How do you make sense of all of this? And I think that's basically the challenge that we struggle with daily with these massive data sets. But it's now gotten a lot richer because of all this other information. So we now know a lot about the distribution of the shape of the dendrites. We know a lot about the synapses that they receive and how they distribute. We now have the information about vesicles, about the mitochondria. And in each case, there seems to be something interesting, a further level of detail that gives us something even more interesting to look at. But a little bit as Guillaume was saying before, all of these structural arrangements are all really interesting, but we need to somehow bring them to function. What does it mean for a dendrite to have these types of mitochondria? What does it mean in terms of how it integrates information and how it functions? What creates these mitochondria, and why is there a distance dependence such that more distal ones contain these weird mitochondria? And I think there's something, we don't know, but then some of these mitochondrial arrangements in the distal dendrites have been observed in more stressful conditions. And so it may well be that the more distal dendrites that are far away from the goodies that are made by the cell body could be under some form of stress, right? And that's the way that they operate. And that might be the normal environment. It could be something completely different. And at this point, we don't really know. But I think that the next level is to be able to see functionally measure things to try and understand what these structures are really doing.
Yeah, But also, yeah, I mean, I have to say that it's a bit like when we were talking earlier about having to upload data roughly. It's kind of a good problem to have. We love to do this and to have more questions
Yeah, it's
to
like a puzzle.
answer.
Yeah.
It's really literally our job to try and answer new questions.
Yeah.
So that's really good. But the other thing is that some of these things can be answered with new experimental conditions, and by having the ability to analyze this, it promotes on us the will to do new experiments and maybe create new data sets, that we if we know that we can analyze them, we can design experiments and create new data sets that
Yeah.
could test our hypotheses.
I mean, like any new,
Yeah.
like any good experiment,
Mhm.
this has thrown open many doors, many new questions. Then it's answered. Those are the best
Yeah.
experiments, without doubt,
Mhm.
because it's basically opened up a vast area that we now need to study. Importantly, and I think this is something that has to do, that is will be one of the areas that we're moving into next. We don't know how any of these things are affected in neurodevelopmental disorders, and there are many of these neurodevelopmental disorders are also linked to a dysfunction of mitochondria, certainly of synapses. And so trying to bring all of these aspects that we have here in a normal brain, if you will, or a standard brain, if you will, it would be really interesting to try and look at them in models of neurodevelopmental disorders such as autism spectrum condition, or epilepsy, even schizophrenia.
Yeah, and I should also add that we're talking about our own research, the research we do in this lab, but I think a lot of these data sets, I mean, our data set is freely available for anyone, and a lot of new data sets are being created, especially a lot of data sets are being created from human tissue, for example. And these tools that Chloe is developing, these new improved detection algorithms and things like that, can be used by other people as well, independently, other research labs.
Yeah,
So that will really help the whole community,
for sure.
and not just
That's
our lab. Yeah.
That's the plan. Yeah.
Yeah, I think it's important to like recognize also the scale of this. We've talked about structure and function a lot, and we talk about it because in biology, structure equals function, right? You can learn something about the function of what it does by its structure. But the other thing we haven't talked about is how varied biology is,
Yeah.
and how one cell can look completely different to another one in health and in disease. And so being able to actually pick up on many, many different observations and quickly collect that data, and then also look at new data sets and look at that too, it just makes it so that you're moving from looking at one example and trying to kind of guess at how the whole world works from that one cell, and look at kind of much more data and get a much better picture about how the complexity of biology works in general, right?
Yeah, no, I think that's a great analogy. It's with like when the first genome sequence was created,
Yeah.
people thought that that was the end, and now there's more and more genome sequences being created. And it's the same with the electron microscopy in general. Yeah, a lot of work has been done at looking at very few examples, and more and more we're
Right.
figuring out. Yeah.
Yeah, it's the whole ethos behind this idea of the connectome, where people are using these large scale reconstructions of these 3D, very large scale 3D EM electron microscopy structures that allows them to essentially create a wiring map of all neurons within a specific brain region. So far, people have mainly focused on the cortex. And they've been doing that, but now the hope is that all of that will speed up not just the acquisition of that data, but also the analysis of that data, which is really, that segmentation is really one of the stumbling blocks, I think, one of the funnels of all of this. And so I think that, as soon as that begins to happen more and more, we're going to start gaining a lot more information of different brain areas, of across different individuals, and now you can begin to compare heterogeneity even across individuals, right? Imagine that, not just within one brain region and so on.
Yeah.
That's really exciting to
Yeah.
be able to do that.
Yeah, and the analogy there with the genome project is exactly that. You know, you started off and it took years to be able to do an entire genome of one person, and now what we can do an entire genome in a few hours, right? So things have really moved fast. And I think that's the beauty, that the amount of information that is thrown at us is now, you know, massive.
It's
and
just that the scientists have to catch up, right? Because you know, they have to now look at all of this data, and it's, you know, just one person, and that's why we have to create these tools, these automated algorithms that can help us to like look at all of that data that it's just impossible to look at with a single person.
Exactly.
Yeah, but also, I mean, with Chloe's presentation is also bring out the importance of proofreading, because I during my career, I've had a fairly long career so far, and we've had throughout my career exposed to many automated methods, and my cynical view is always that it looks good on the examples that they have, and it can do some automation, but the proofreading time that's required to make it be workable and analyzable, it takes longer than if you were to do the segmentation by hand. So if the proofreading time is not there, it's really a big advance, and it's very important.
Absolutely. I think we're probably running out of time, but I think unless there's anything else that you guys think we haven't covered, I think it's I think we've kind of gone through the major milestones and the major results, haven't we? Yeah.
I think so.
Okay, well, fantastic. Then I think we will end the stream here. One thing that I definitely want to say is that if anyone's watching who is, you know, contributed to the project on the Zooniverse, we are so eternally grateful for all of your hard work. It's given us a lot of work. I'm pretty sure the time that the project was on the Zooniverse is less than the time Chloe's had to spend analyzing all the data, and far less than all of the kind of research that's going to come afterwards. But as Juan and Guillaume said, that is a good problem to have. So we are very, very happy.
Thank you, everyone. It's been a massive help.
Thank you.
Thank you. So I'm going to wrap up here. I'll be back on the 12th of December for the next live stream, Science Scribbler Q&A and Classify with Michelle. But thank you to Juan, Guillaume and Chloe for coming along. And also, thank you to our Science Scribbler community and to the people who contributed to Synapse Safari. And we'll see you next time.